The next speaker is Dr. Shota Hatakeyama. Is the last but not least. So Dr. Shota Hatakeyama is a certified senior embryologist and laboratory director at Janayara Women's Clinic. He's a prominent expert in reproductive biology. He holds a PhD in medical science from Gunma Path University in 2021 and a bachelor's in agriculture from KITAZATO University in 2007. Specializing in assisted sperm fusion insemination, ASFI, technique he developed. Hatakeyama has improved fertilization rates and reduced oocyte degeneration compared to the conventional ICSI. His work on ASFI, published in Fertility and Sterility and presented at major conferences, has gained international recognition for advancing fertility treatments outcomes. Today, Dr. Hatakeyama is going to talk about this recent study on the alternative technique to the conventional ICSI and its promising clinical results. So please, Dr. Hatakeyama, we are ready when you are. You can start.
Thank you very much for the kind introduction. Good evening from Japan. First of all, I would like to express my sincere gratitude to北里コーポレーション for organizing this wonderful webinar and to Andrew for moderating today's session. I also truly appreciate all of you for joining today. So in this presentation, I would like to introduce a novel micro-insemination method called ASFI, which aims to overcome some limitations of ICSI. In 2020, we published that fertilization was obtained by pressing a zona-passed-bound sperm onto the oocyte membrane. We designated this novel micro-insemination method as Assisted Sperm Fusion Insemination, ASFI. So you may wonder, how did we come up with this method? About 17 years ago, when I started performing ICSI clinically, I began to wonder if it was possible to fertilize oocyte without breaking oocyte membrane. Then I recalled a micro-insemination method known as SUSI, and I wondered if it could provide a less invasive approach to fertilizing oocyte. However, it raises the question, why was the fertilization rate of SUSI so low? SUSI is a micro-insemination method that involves injecting sperm into the perivitrin space. The official name is subzonal insemination, and it was introduced in 1988. I speculate that the low fertilization rate might be due to sperm moving freely within the perivitrin space. Based on this speculation, to prevent sperm from moving within the perivitrin space, I attempted to press the motor sperm head against oocyte membrane using an injection pipette for about 1 minute. Then, after removing injection pipette, I observed the sperm head remained attached to the oocyte membrane while its tail was still moving. At the time, I thought I'd discovered an excellent method. However, when I performed the same method with other sperm, it did not work. Even though I extended the duration of pressing the sperm to 5 minutes, the result was still unsuccessful. Consequently, it only worked on the first attempt, which made me think it was just a coincidence. So at the time, I decided to give up and did not pursue it further. 10 years later, when I enrolled in graduate school, I decided to revisit the method. But first, I needed to ask a fundamental question. What is the difference between sperm that attach to the oocyte membrane and those that do not? To better understand factors that enable sperm to attach to the oocyte membrane, I first reviewed existing literature. As a result, it was reported that all sperm that attach to the oocyte membrane have undergone an acrosome reaction. Based on this, we hypothesize that the sperm bound to the outer layer of the oocyte, known as the zona pellucida, might also undergo the acrosome reaction. Therefore, we retrieved several motor sperm that were completely bound to the zona pellucida using an injection pipette and investigated the acrosome status. As a result, the acrosome reaction rate of sperm bound to the zona pellucida was 98%, while that was a control group, which referred to sperm not bound to the zona pellucida, was 28.60%. A significantly higher acrosome reaction rate was observed in the ZP sperm group. So based on this observation, we speculate that it might be possible to fertilize oocyte using zona pellucida bound sperm, potentially preventing oocyte degeneration. To test this hypothesis, when we press the zona pellucida bound sperm against oocyte membrane, they successfully attached. Up until that point, we have only succeeded once, but using zona pellucida bound sperm led to success. So we confirmed that attachment was possible. But the next question is, will fertilization occur afterward? Moreover, for clinical application, how can we reliably secure zona pellucida bound sperm? In the next study, we focused on oocyte after conventional insemination to obtain zona pellucida bound sperm. At our clinic, we perform rescue ICSI on unfertilized oocyte after conventional insemination. Therefore, we performed ASFI on these oocytes instead of rescue ICSI. We defined this process as rescue ASFI. Among the patients who underwent oocyte retrieval and conventional insemination during the period shown in the slide, rescue ASFI was performed on 89 couples who consented to participate in this study. In contrast, rescue ICSI was performed on 233 couples who either did not consent or did not obtain zona pellucida bound sperm despite giving their consent. As a result, rescue ASFI produced comparable fertilization and blast formation rates compared to rescue ICSI. Furthermore, rescue ASFI exhibited significantly lower degeneration rates. However, a risk of polyspermy was observed when performing ASFI instead of rescue ICSI. Moreover, it is not technically clear whether the oocyte was fertilized by sperm pressed against oocyte membrane or not. To address these concerns, performing ASFI on oocyte without conventional insemination is necessary. Therefore, in the present study, we aimed to investigate whether ASFI without conventional insemination can enhance fertilization and embryonic development compared to conventional ICSI. In the next study, we utilized immature or degenerated oocyte to secure zona pellucida bound sperm. This prospective study was conducted on sibling oocyte. The study was approved by local ethics committee of our clinic. Patients with inclusion criteria of retrieving at least 2 metaphase 2 oocyte, 1 immature or degenerated oocyte, and the absence of severe male infertility conditions were recruited. Consequently, 24 couples undergoing 34 oocyte retrieval cycles were included. Written informed consent was obtained from all patients. After oocyte retrieval, cumulus oocyte complexes containing metaphase 2 oocyte were incubated exclusively. In contrast, seal seed containing immature or degenerated oocyte were incubated with 10,000 motor sperm. After incubation for 2 hours, the cumulus cells were removed by pipetting. Subsequently, these oocytes were incubated with sperm for an additional hour to enhance the number of sperm bound to the donor pelster. In contrast, the cumulus cells surrounding metaphase to oocyte were removed using hyaluronidase. Subsequently, metaphase to oocyte were alternately allocated into conventional ICSI and ASFI. If the number of metaphase to oocyte was odd, the surplus oocytes were assigned to the conventional ICSI group. In the ASFI group, after aspirating motor sperm bound to the donor pelster, the sperm was pressed onto the oocyte membrane. This slide shows a demonstration of ASFI. The donor pelster-bound sperm was already aspirated into the injection pipette. The pipette was then used to penetrate the donor pelster. Subsequently, the sperm head was pressed against oocyte membrane using the bevel of the injection pipette for 10 seconds. Finally, the injection pipette was gently removed without interrupting sperm motility. After removing injection pipette, we observed the sperm head remained attached to the oocyte membrane with its tail moving. The tail movement gradually stopped. Using a time-lapse system, we were able to observe sperm incorporating into the oocyte. The sperm is attached here. As shown, the sperm head gradually incorporated into the oocyte over time. This slide shows fertilization outcomes. The two-pore nuclear rate of ASFI was 18.8%, while that of the conventional ICSI group was 17.2%. A significantly higher two-pore nuclear rate was observed in the ASFI group. Moreover, no oocyte degenerated after ASFI, resulting in a significantly lower degeneration rate. This slide showed outcomes of embryonic development. There are no significant differences observed in the rate of good embryo formation day 3, blastocyst formation, and good blastocyst formation between the two groups. However, the usable embryo rate was significantly higher in the ASFI group. So, based on these results, ASFI has several advantages. The first and the most remarkable advantage of ASFI is its ability to fertilize oocyte without breaking the oocyte membrane. In ICSI, despite the case of refinement, including the introduction of the PACE method, it has remained extremely difficult to eliminate oocyte degeneration, since the process inherently involves membrane disruption. But in our study, not a single oocyte degenerated after ASFI. This is a groundbreaking result. ASFI has overcome one of the most critical and persistent limitations of ICSI, oocyte degeneration. The second advantage of ASFI is that only sperm are incorporated into the oocyte without additional substances. In ICSI, substances other than sperm, including a media, PVP, and coarse enzymes, may be injected into the oocyte. Although the effects of injecting these substances into oocyte are not fully understood, these substances should be avoided as much as possible. Whereas in ASFI, there is no risk of injecting unwanted substances into the oocyte. Thus, while ASFI resolves some of the disadvantages of ICSI, it is important to acknowledge that this method still presents certain challenges. The first challenge is the requirement for the donor pelster. ASFI cannot be performed in cases where immature or degenerated oocyte are not available. Therefore, to expand its applicability, we need an alternative sperm selection method that does not rely on the donor pelster. Second, this study used a small sample size, more extensive studies, including pregnancy outcome data, are required to verify our results. So to summarize, ASFI yielded significantly higher fertilization rate and a significantly lower degeneration rate compared to conventional ICSI. It increases the number of embryos available for implantation. Although ASFI presents some challenges, this novel micro-insemination technique represents a viable alternative to the existing techniques for terminal infertility. That concludes my presentation. Thank you very much.
Thank you very much, Dr. Hatakeyama. Very good presentation, great work. So it's really curious how a technique so established until today can be slightly modified and significantly improve the current results. So thank you very much and really nice work.